So this is the second half of our lecture on heterotrophic protists, so things that used to be classified as fungi but no longer are. We covered the oomycetes last time. This time we're going to look at the myxomycetes and the dictyosteliomycetes. These are both types of slime molds. We have the cellular and the acellular slime molds. And then the last part we're going to look at the big phylogeny. So kind of starting to think about how all of these organisms are related and what a phylogeny is. So the body plan for the myxomycetes is nothing like fungi. They are forming something called a plasmodium. And so that plasmodium is like a giant multinucleate, so many different nuclei, amoeba. And when they make swimming cells, those cells have two flagella. Whoa, that looks pretty crazy. I can't write fully on the right-hand side of the screen, or I press a button and then it changes the slide. So they have two flagella, and those flagella are just normal, whiplash flagella. So environment, where do they live? They're terrestrial. You're going to see them mostly in moist soil or decaying wood, these wet environments where they have a lot of stuff to eat. Energy acquisition, they are heterotrophic by engulfing. So whereas fungi and our oomycetes are going to be sort of barfing out these enzymes, digesting their food on the outside, then absorbing it, our myxomycetes are going to travel around as this big blob, engulfing their food. Their storage carbohydrate is still glycogen. Their cell walls, when they're present, are made of cellulose, but they only have a cell wall on their spores. Ecological function, they are saprotrophs. They eat bacteria, which I suppose aren't actually dead, so they're consumers. Okay, so our big differences here between the myxomycetes and our true fungi is that weird body plan that they have with their two flagella when they have them, cellulose in their cell walls when present, and that they engulf their food instead of digesting it on the outside. So here's a review of that information we just wrote down. Just in case you missed it, you can go back to this slide and look at it. Here's a picture of the Physerum polycephalum life cycle. I just took pictures of the different stages. So this is a myxomycete. It's probably one of the most studied myxomycetes, if not the most studied. And so we're looking at its life cycle, which is a diplontic life cycle, which is important because that's another big difference. So you have a diplontic life cycle, much like the oomycetes, but unlike the fungi. So up here, this is our haploid stage. Meiosis would be happening inside of these structures to create spores. That, I guess these aren't really spores in that they don't grow. They just sort of insist and then germinate, and they germinate into these swarm cells. And you don't need to memorize this life cycle. This is just for your interest. So these swarm cells are these little amoebae that swim around. They can take on a few different forms. They have two flagella, and then they find each other and they fuse together, and then you get this diploid plasmodium that's then going to grow as this giant amoeba and seek out food. And then once it runs out of food, that is going to trigger it to make spores. This is a picture of an experiment someone did on Physerum polycephalum. So they arranged these oats, which is something that Physerum likes to eat. So if you want to grow this at home, you can feed it oats and it'll grow on your wet paper towel. So they arranged these oats in these major locations for the Tokyo subway network, and then they put this cube of Physarum polycephalum on whatever it was eating right in the middle, and they wanted to see how accurately or how it would sort of map these locations and connect them together. Because as it spreads out, it's finding food sources, and then once it finds those food sources, it makes a connection between its last food source and this new food source as a way to sort of piggyback around this environment, always finding food sources. So it made a pretty accurate map of the Tokyo network, and it's a really interesting, not fungus, but fungus-like organism to study for its ability to sort of solve puzzles like mazes by this food locating ability. So here's a bunch of slime molds. These are all in the Myxogastria. They are diverse in appearance, but they all have that same strategy where they're initially this slimy plasmodium crawling around, eating bacteria, and then once they are triggered to make spores, they make these very different looking spore making structures. So the one in the top left you might have seen around, that's Phylogenetic, I believe, and it forms this hardened white crust around it, and eventually will make brown spores. And that one's called the dog vomit slime mold. Kind of just looks like a pile of mustard hanging out in the forest. But you could see, I think, all of these in our local woods, but they're very small. You got to be looking at that centimeter or less level. So the next group we're going to learn about is the dictyosteliomycetes. These are called the cellular slime molds. So our myxomycetes were the acellular slime molds because they're not really made of cells. They're just one big flobby amoeba. But here we have many individual cells. So individual cells that are not the same organism, that when they get this environmental signal, one of them starts sending out this compound called cyclic AMP, and that causes all of the other ones around it to aggregate toward it. And so then they all start emitting the signal, and so then it causes all of them to aggregate into these clumps. And once they aggregate into one of those clumps, then they start moving around as if they were all the same organism. So this is a video that you should watch. I'll link it in the comments. It's very fun and the narration is very good. So this structure here is an aggregate of a bunch of those unicellular dictyosteliomycetes. And they clump together in this structure called a slug or a grex, and that moves around following sunlight because it's going to try to get up to a place where it can disperse its spores. And if you think about it, these are all different organisms, and they are going to make one structure, one sporangium, and that means that a bunch of them have to end up in the stalk of that sporangium. And those ones aren't going to become spores. They're going to die in that stalk and not reproduce. Only the ones that form the spores are the ones that are going to reproduce their genetics. So this particular group is studied for altruism, the kind of selfless giving to promote the existence of the rest of their, or survival of the rest of the species. But interestingly, there's always cheaters. There's some certain percentage that will always make it as spores. So interesting group of organisms. Okay, so a phylogeny. That's a new term maybe for a lot of us. So throughout this course, the rest of this course, we're going to be building a big phylogeny of all the organismal groups that we study and adding traits on that. So first we have to learn what a phylogeny is. So a phylogenetic tree is a way that we can visualize the evolutionary history of organisms. So, we drew a phylogeny for kingdom fungi, the really hokey one, right, where we made this tree-like structure. And really, if we don't put distance for evolutionary groups on it, it's just a cladogram, I guess, where we're saying that, okay, here we have 3 different groups of organisms, and they all stemmed from this shared common ancestor. So at some point, this organism reproduced back here. This node represents an organism that reproduced, and then its offspring evolved in different pathways. Some of them would continue on to be the ancestors of species A. Other ones would go on to be the ancestors of the rest of these species, B, C, and D. And at each of these points, we have some major change that causes speciation within this group. And so that can be just about anything, but it has to be some barrier to mating. So the offspring are going to have different characteristics, and maybe they end up on different continents due to drift of those tectonic plates, or maybe one has a mutation that doesn't allow it to mate with the others. So it can happen in a lot of different ways, but we get some mechanism that causes divergence, and that causes branching in this tree of life. So the phylogeny itself, that tree that we build, is just a hypothesis, how we think that those organisms are related, how we think that their evolutionary history happened throughout the course of time. But we don't know for sure. We build these hypotheses based on evidence, as you do with any hypothesis, but the evidence that you choose to incorporate within that, you'll always be limited in what you can include, and if you try to include all that evidence, you might get a completely unresolved tree. So all of your branches kind of stem from the same point. It has no ability to tell you because there's just too much conflicting evidence, right? And so if you look at different groups of evidence, like some important gene and the changes in that gene throughout time, you might then look at another gene and the important changes in this other gene throughout time, and they each tell you a different story. So as a scientist, you have to decide which of those stories you're going to believe. And so that's going to be based on the evidence you have at hand. It could be genetic, it could be morphological. For any good phylogenetic tree, it's usually going to include a wealth of information from all different aspects. So depending on that information, you might get trees that lay out differently. And so the phylogeny that we'll build is going to be based on new information that was just recently published using, it was like 1,000 different transcriptomes. So looking at all the different groups of plants and the genetic products that they're actually making within those organisms, and seeing how all those products have changed throughout time. And so that's the base of the information that I used for how we're going to draw our phylogeny. But I'll show you two different ones that have representation in scientific education. So here's one from 2005. This one's pretty simplistic, but it's very similar to the one that we'll draw. So we have the green algae broken up into a few different groups, and how those resolve is still pretty similar, but not quite the same. We have the non-vascular plants, so those are our bryophytes. And if you look at the order they're shown on in this phylogeny, it shows that liverworts came out first. Right, so here's our liverworts, and they have a different name now. They're the Marchantiophyta. And then it goes hornworts, then mosses. So remember that order. Liverworts first, then the hornworts evolve, then the mosses. The other difference here, that'll be different from the next one we see, is that our gymnosperms are unresolved. They're all emerging from the same point, so this just says, based on this information, we don't know who came out first. We don't have some order of descent. We have this unresolved part of the tree. So you can compare that to this one, which is excluding the green algae, which most things that are talking about plants now actually include parts of the green algae because they're so closely related that to call them, think of this group as the plants, you actually have to include them. So here we have the liverworts coming out first, and then we have mosses, then hornworts. So in our last one, the hornworts and mosses were switched. And this happens a lot. It's really difficult to find consensus on how the bryophytes evolved. In the information that we're using for our class, most of the evidence actually pointed to the bryophytes all sharing one common ancestor. And so that is completely new and different from all of the other information that has been out there for a while. So it'll be different in your book. They'll probably show it in this order, where you have liverworts, mosses, then hornworts. You can also see that the gymnosperms have been resolved. So we have the cycads coming out first as our oldest lineage of gymnosperms. Then we have the ginkgales and conifers and neophytes sharing this common ancestor here. Interestingly, for what we're going to learn about, it looks like genetically that the neophytes might actually be within the conifers and be most closely related to the pines. So that's all just looking at a bunch of different genetic information and seeing what gives you kind of the best, most well-supported outcomes. So we'll see a lot of different types of trees, both plants and phylogenies, but they'll all be different from each other because they're using different information. That doesn't mean that one is a good tree and one is a bad tree. They're all just different hypotheses, and we are either gaining or losing support for those as we gather new information.